Thanks for inviting me, and then thanks to the organizers too. Sam was telling me a little bit about the organization, so it's a good initiative, and I think we need more of that going forward. So, like he earlier said, I try to acknowledge my beginning. Started from Michael Obara University of Agriculture, and then proceeded to Cornell University in 2013. And I would like to say that though I'm working with Bayer, but what I'm presenting today, I don't have the permission of Bayer to speak on behalf of Bayer. So what I'm doing is not what we do in Bayer. And I was asked to present some of the work I've done before now. So it's nothing related to Bayer. And if you want to know more about Bayer, we can talk about that, or you can reach out. So today we are looking at digital eyes on African fields, which is high throughput phenotyping, and then for the next generation crop improvement. Quickly, I believe so much in agriculture and the potentials we have in Africa. It touches the economy, and in some countries, agriculture contributes to about almost 40% in some nations, if not more, in their GDP. And what it means is that it's so important in the African context. And we have most of the African land that are uncultivated. So what it means is that we can change the history of Africa if we can capitalize on the potentials we have in agricultural sector. If you look at data and statistics, sometimes they don't agree with the reality, but then we are hoping that part of these initiatives and then challenging ourselves will be to change the narrative. When you look at food and nutrient deficiency in African context, is a little bit demoralizing, because the estimate FAO believes that more than 220 million Africans are undernourished. Yes, we can argue about the data, the source and all that, but if we look deeper, we need to also produce, if we want to probably debate over this, we also need to do more, and if possible, have more data to disprove this. I know people might be like, okay, where and where? But the reality is that a lot of Africans, and when you look at undernourishment, it touches in different aspects: vitamin deficiency, iron deficiency, zinc and protein. There are so many data points. If you look at also hunger map of the World Bank, you realize that most of the areas in Africa are really, when you look at the color chart, the red and the yellow are more of places where we have both climate and natural disasters and things that can hinder the production of agricultural production. So most of these crises are mostly in rural areas where a lot of people are at risk of food and nutritional deficiency. So you and I is a call for action, and that leads us into the issue of phenotyping. Why are we talking about phenotyping? What we are trying or aiming to do is to increase or improve food production. We want to improve the genetics of plants, produce more yielding, more disease resistant, more drought tolerant varieties. And what role does phenotype play in this whole context? Phenotyping, I was told that some people don't have the breeding or plant phenotyping foundation. So we are talking about, what can I measure? So it could be height. And why is that important? In my attempt to produce more varieties, I need to measure if I'm breeding for yield, I want to look for a way to measure my yield and be sure that it's high yielding in the midst of large population. And then, so it could be height, I'm improving disease resistance. But overall, why it matters is that both my gene and my environment interact. So if I can develop a mechanism to measure things effectively and then accurately, that is why it matters so much in plant breeding. So it provides the data we need to select plants, that's in summary. And it plays a vital role in trait identification. If I have a large population I'm screening, I need to be sure that the individuals I'm selecting have the trait of interest. And we use phenotyping a lot, especially when we're trying to track or link whatever genotype we have to the phenotypes, because overall, the new genomics or direction of breeding is that we're using genomic selection, we are using or genomic assisted breeding. So we are looking deeper than what the physical eyes can see. So what we need to do is that we need or have a means of phenotyping and phenotyping accurately. So that is where high throughput comes in. There are a lot of groups that will ask you, when is phenotyping high throughput? Is it only when you have drones, sensors, and talk about AI? It can vary. You can look at the scale, ability to measure in a large scale. You can look at precision, ability to measure and then be precise and know exactly what you're measuring that is correlates with the truth. And you can look at, yes, the drones, the sensors, all those things can come in all together to play a role in high throughput phenotyping. Some other people can say, is it fully automated? Yes, even before you get to the automation stage, you also need to build up, and that will lead from calibration and all the rest. So one of the things I've been asked to or use this forum to do is to open our eyes to possibilities in high throughput phenotyping, looking at case studies in some of the things I've done, and some I directly led and some other ones by partnership in the lab or so. But I think we'll get back to what are the challenges, and then looking at it from African context, what can we do moving forward? So the first case study will be some of the work I started in cassava, and this work was done under the Next Gen Cassava project. It lasted for like several years, over to like different stage one and stage two. But currently, what is cassava? Cassava is a major source of food to a lot of people. It's not just vital in Africa, but across the world. South America, people use and eat it in different forms. It's one of those crops that if you are from Africa, Nigeria is the leading producer of cassava, but then it's something you'll appreciate because that is what people rely upon. Although maybe today is changing, it's no longer the crop for the poor. Is both for the poor and for the rich. One challenge is that cassava has a long breeding cycle. So we are looking at above between 8 to 10 years. It could be more to release a variety. And why does that? The implication of that is that the people are looking for more yielding variety. We are confronted by a lot of challenges in climate. So if we need to respond to some of these requirements, we need a way of increasing that breeding cycle or producing more so that we can meet the demand in the market. So the traditional phenotyping in cassava is mostly tedious. It's limited with a lot of amenities. If we're looking at extraction in the lab, most of them will require technical knowledge. The scale is not as we want, especially in the era of genomics assisted breeding, where you need a lot of materials for screening, and then you need multi-locational experiments and all that. So the implication is that it's affecting our prediction, and some of the methods, as I go deeper down, you will see that some of them might be biased and prone to error. And that has implication in our prediction accuracy and genetic improvement. I don't know if my highlighter is working here. So I want to break it down a little bit, trade by trade. So I will highlight some of the trades just to trace the point I made earlier. If you are measuring dry matter content in cassava, there are two methods we have, the oven drying method. And the approach is that you just take like between the 3 to 5 kg of your sample, you peel, wash. This method is the method that has been like what was originally like established for dry matter content evaluation. You dry it, so dry matter content, you're just looking at the mass minus water, dry content. So that is where you base on your yield on, and then you can equate or relate every other component you want to think about in that yield equation. But the problem is that you are looking at a lot of samples, like I mentioned, for us when we started in genomic selection, we were thinking about training at least 1,000 materials or varieties in different lines, in different environments. At least 3 environments was our target, and then different locations. So you have your rep, you have location. So when you multiply that, you know you are talking about a lot of samples. So it's completely inadequate for such large population, and then highly dependent. You are thinking about oven drying that will be steady for that 24 hours in some part of the world, in Africa. That might be impossible. So another method that is more like prosy to dry matter oven drying is the gravity method. It's about the same thing. We are looking at displacement in water. So you have to weigh in air, take a sample, you weigh in air, and then weigh in water, and then look at the ratio between the weight in water and the weight in air. And that gives you the gravity. And we had some earlier research that gave us like a relationship you can use to calculate your dry matter content and starch content, because both are related. It's time consuming, labor intensive, like I mentioned, and then you have a lot of variation. The practical aspect of it is that there are a lot of things compared to where you have your oven drying, where you peel and take out some of the stock. If you are close to cassava root, a lot of things can cause a bias in your data. So we were looking for an alternative that can help us get something quicker, something more predictable, and then scalable too. So the last among the traits I will mention here is carotenoids. We're breeding for high carotenoid content, vitamin A. At a point, we could only concentrate on total carotenoid because we didn't have the facility. If you are looking at quantitative method of extraction of carotenoids, you should be looking at HPLC, where you can get all the, not just total carotenoid, but you're going to get all the components. But for us, where we have that large kind of large scale of production, and then we're trying to meet our target, we had a lot of ratings, but the most consistent one would be the scale of, you're looking at white, cream, or yellow. But then when you are looking at this picture here, you look at the variation in yellowness can also be a bias. So when you are scoring, and depending on when you are scoring, if the sun is out, the reflection of the yellow I give might be different from my neighbor, and it calls for a lot of bias. So pure white, creamy, do I now categorize this as is quite creamy 2, and then 3? At some point, we're using a scale of 1 to 5, where 2 and 3, people cannot even like differentiate between those 3 in between ratings. So we could have bias depending on who is rating, at what point in time you're rating, and all that. So an alternative would be going towards precision, looking at some high throughput options. And spectroscopy was one thing that we explored. We had a desktop format that was being used as CADs, and we had potentials to check what was in vogue, advancement in technologies, and we started looking towards the portable version. And I don't know if I need to go deep here. What you see X-rays, UV, IV, and what we see could be in different ranges. So what we are trying to capture is beyond what the physical eyes can see. And once you have your matter or your sample interacting with electromagnetic radiation, you usually provide results in wavelength, and we want to use that wavelength. So that is all about spectrometers and spectroscopy. It covers most of the tools and technologies you have in high throughput phenotyping. Use of sensors, most of them are based in this technology. So we went for a portable format. Usually, I think initially when the producers told us that it was mostly for mining, but we agreed to test it for sampling of food. So the process is simple. You go from sampling, acquire your spectral data, like I said. Once the electromagnetic wave interacts with what matter you have, it produces a wavelength. And so you get your wavelength, you have some pre-treatment you go through, and those might be just to correct for type of sample and different things, and then light dispersion and all the rest. And the building calibration, so this is more like calibration protocol. Like I said, the foundation is about the same, whether you're using hyperspectral or some other sensors. Most of them are based on the same technology or protocol. So the next thing you need to do is you have a reference, and that reference could be what will be the source of truth. And for us, the dry matter oven drying is like the best ideal source of truth for dry matter content, and for carotenoids, you need quantitative measures from HPLC. So the two things you need to do is to develop a model. You can use a lot of models. We can talk about that later. I didn't go deep into that. What you can use, your machine learning and different linear, non-linear. So there are so many options you can explore. Then you want to build a calibration that will cut across different samples, and then you test and validate your calibration model. So you have your training, will be the samples that have both spectral data and then at the same time have some reference information. You do that and then create and validate that equation before using it. What we are doing is that at the end of the day, you have a lot of samples because you are limited here. There are few samples you can get the reference values for. So at the end of the day, you want to develop a relationship between the spectral data and your reference value, and then use that moving forward to predict samples that have spectral data, but they don't have weight chemistry. You can now predict it using this equation you have established earlier on. Sam, I think questions will come at the end of the day. I don't need to pause because of time.
Yeah, you're correct. You're right.
So I just want to quickly show results of what we did, our initial attempt. Like I said, there are so many considerations. What we measured on it mostly was how does our equation or our results compare when you are using intact route, because that would be our ideal thing. You are in the field, you are far away. I think we did this in Kano, in Nigeria. And the distance between Umudike, where the institution, some of these, I think we had two institutions, we had branches, though. Michael Opera, National Rural Research Institute, close to Michael Opera, and then IIT. We did that in collaboration. But then the distance between those two sites and Kano is almost, you're talking about a day's journey. So we want to do something that we can do in the field, because you don't want to carry this sample going back. If you're using oven method, these are like experimental fields where you don't have other facilities. So we are using them for testing and trial. So our ideal thing would be to scan in the field and not carry anything back to the station. But then it's possible to scan in the route But when you look at the root development, just looking at this, the development from the stalk to the tip, you realize there are variations for so many traits, and there are some research that have proven that you have variations. So the option is to make sure you crush this and have a good sample of your roots, sampling of your roots. So we talk about, do we mash it or do we just do what will be the result, correlation between the result when you develop this calibration on intact root and then when you do it on mash. But so far we got some good equations or correlation between our prediction and then the real samples. If you are used to this, I might not go to breaking them down, but in some instances, they were good enough, even in the intact roots, to give us the accuracy we need to proceed with selection and screening. And for dry matter, for carotenoids, we have we had the same results, both intact and then using the mash samples. And to some extent, looking at total carotenoid content and then all beta-carotenoid content, they were all high. Not just that, we went further to get different components. That is one advantage, like I said. Earlier on, we were just looking at total carotenoid contents, and that was based on color rating. We're assuming that the more yellow it is, means it has more carotenoid content. But that didn't give us enough room. But now, having this, we're able to break down into different components, because you talk about carotenoid, there are different lutein, and even the beta-carotenoid content has both C and 13 and 15 C's version of it, alpha-carotenoid. So that was opening a lot of space for us, expanding our phenotyping capability, and then giving us room to really be precise and know what we're breeding for, because based on total carotenoid, there are so many other things that are hidden within that we couldn't account for. So the value of that is that, like I mentioned, we're able to check some of the genetic correlations. We're able to do GWAS, do genomic predictions on some of these trait components. Going forward from there, I think this should be the first postdoc doctoral position I took on, was looking at plant innate ability. So plants respond to differences in the environment, to different stressors. It could be drought. While it might not be visible to us, they would have started responding to it. The Asian people might use differences in color or change in color to predict that weather is going to change. For that is when it's already like extreme. But we believe that some of these reactions or interactions, sensitivity, they start as early as possible when those environmental changes happen. So it could be biotic or abiotic. So what we're asking, can we study and understand those signals from plants and use them in plant improvement? It could also be in detecting drought, in detecting early detection of drought or some of the stressors in the environment. So moving from, like I said, I'll be touching on different technologies. So mostly we base this on hyperspectral imaging. And I might not go deep into trying to explain the variation in the same wavelength, but then we are looking at different dimensions of spectroscopy, and then going into a lot of pixel and developing calibration for that, so that we'll be able to predict those variations. What we did was we set up a few experiments. We had some vegetative indices that we use to separate between water stress plants and non-stress well-watered plants. And yeah, we were able to, at least based on some indices and wavelength region to say, okay, we'll be able to use this moving forward. I contributed at the early stage of the project. It was more than this. I might not be able to get more scope to that, but these are possibilities. And the other option was using thermal imaging. So we tried using different vendors. We started with Flare, but then I think we moved forward to develop some Python models so that we'll be able to capture and differentiate between differences in temperature. If we scan the plant, we'll be able to show the ones that are stressed and the ones that are not stressed, the difference or the variation, just using thermal imaging. At the same time, we had some component of transformation we were doing to enhance that ability of plants. So we had some plants that were transformed to have higher expression of the now this was the thermal or resistance to drought. So were we able to capture between those two genes? As you can see from the graph, in most instances, we realized that we had, and we kept also improving depending on, so it was an ongoing experiment. We do a transformation, we look at the level of expression, and then know whether we're increasing the gene frequency so that we could increase that ability to separate between the two classes. So yeah, that was possible using thermal and also the hyperspectral combination. I will run to computer vision, which is also a component of high throughput phenotyping. Like I said, some of these technologies are based on electromagnetic spectrum, what you see, the visible light, the infrared, microwave. There are so many other things going on that you and I cannot see, but using these devices, this hyperspectral is that we have multiple wavelengths we can capture. Multispectral is like we have a few limited bands that we can capture, and then RGB will just depend mostly on what the eyes can see and the region of visible light. And you can also break down into the red, green, and blue regions. So we did some work on graves. That will be my second project that I did at Cornell. And so what we did in terms of automation, because nobody can carry this and be imaging the whole vineyard. So we got this motor that we can mounted. This involves a lot of, maybe I can mention here, a lot of interaction with vendors to know. Some we will test and then we will burn the bulbs, or the bulbs will burn within a day, within an hour, and then you look at another vendor, we look at different things. So it involves a process. So that is also something maybe when we are talking about the challenges to bear in mind, that it might not just be one model fits everything. But this we're able to capture, like we can use a TV run and mount it in the field. So this was helping us capture images in the field, and we're trying to calibrate for leaf diseases in grapes, which was so devastating in the northeastern part of the US. So far we're able to generate a lot of, the process for this involved annotation. So the first thing, more like your reference, is for somebody to first sit down, do the annotation, do the rating, and then we feed it into the model so that we'll be able to automate the process. You don't need somebody each time to go in there and do it. You just feed it into the process, and then you'll be able to predict the rating of the disease. So we're able to we started it, kicked it off, and then so far I think that project is still ongoing, and we have some good results that we can use not just for phylloxera. We also have some other diseases, agronomic traits, and that we went further into. Before I left, we were trying to predict leaf flower characterization, when the flower initiation, budding in grapes and all that. Those are things you need to go from one vine to the other, or one line to the other to score. But if you can have that and automate them and calibrate them, because sampling, like I said, in cassava, maybe a plot of 10 by 10 meters, you are taking a sample of 3 kg. If you are scoring the whole of this vine, you are limited to 1 or 2, and that can vary. But when we have a larger scope, we'll be able to, and the system is trained to do that, we'll have a larger, higher accuracy to predict some of those traits. The other thing we did, got exposed to was, this was during the COVID area, so we're all masked, was to look into fruit quality, especially for non-vinifera grapes. We had some model that people were developing, but for wine grapes and some other grapes in the US, we needed to build more equation to develop our own model. So I have been mentioning some vendors, tools. This is not advertising for this, but some of these are some of the things we use. We tested some we dropped, some we continued with. In some instances, we pair between instruments. So the idea was to correlate some foliar properties to sensory attributes of the grape wine, either the wine or the fruit itself. So yeah, we developed some of the data. We are publishing some of the publications. You can check and then see some of the things we've done so far. Some of the data are still there, and maybe that could be a call for action. People who are free. I have accumulation of data I'm still like trying to process. I'm happy to work with anybody that has that skill. So I will touch on some other things. Like I said, some of these were fully under me, and some we developed in collaboration with other folks. So this was in collaboration with the engineering department. This is an engineering student in the same vineyard that I showed earlier. So this is just after the winter. They are beginning to bud, and we are trying to calibrate using the ground robot to score for early flowering, budding, and all that. If you follow Cornell, Geneva, you will see a lot of those development and advancement. So the result is there. So this is just when we started, you could give the dimension and all that, give the field map. So this will be able to run through this whole field and come back with the result, and you have the data immediately. And with the calibration, you'll be able to make your predictions and know for each of the traits you are measuring what it looks like. I will touch on some of the other things we did both in corn and, like Samuel said, my last call before joining Bayer was on soybean breeding. So we were flying drones. I think that is more like the most, when you think about high throughput phenotyping, we think about, oh, we need to fly drones. That is okay. We had these coupled both cameras, and then we were looking at potentials of hyperspectral and thermal cameras mounted on this. So we're able to extract both vegetative indices. We also had some other stand counts. Currently in soybean, we are using it for stand count. Nobody's going each plot by plot to count number of stands in the field at the early stages. So, and some of those indices we incorporated into genomic prediction and all that. These are some of the extension of both the thermal hyperspectral, and then there are other extensions with maize or corn, where they planted sensors into the stem of the growing plant that will be able to give you signals. You can look more into my Gauss lab. You can have more information on that. So I think enough of maybe, so these are some of the possibilities in high throughput phenotyping, reference to some of the things I've done or experienced, but then moving forward, I think the challenge is how does that apply to the African contents? Like I said, we have a lot of opportunities, opportunity to develop this technology. Sub-Saharan African alone, the annual stress or climate-induced stress, and we have losses up to 30% crop losses, and then there are different metrics. African Development Bank estimates that is up to 7 billion that we lose in crop value due to pests and diseases. We have droughts. We have some parts of Africa where people can't really grow crops like throughout the season because of drought and all that. But that also presents an opportunity. Let's not just see it as a challenge, but it's a unique opportunity. I've mentioned that we have the landmass, 60% of African arable or area it can be used for agriculture, and we are underutilizing that potential. Some other, when you compare it to some other countries, India and other places, we have more advantage. And the question of food security and income in some African countries, people are solely dependent on agriculture for their livelihood. And like I'm saying, I have a first-hand experience. I grew up in the village, so I know how people depend on agriculture for their livelihood. Barriers to adoption, I think I've touched on this during the presentation, is while we look at initial costs, at the same time, I can argue that it's not purely a disadvantage, because when you compare this with establishing a functional laboratory for weight chemistry is somehow less expensive to establish. So, but then I don't think we need to shy away from the fact that it's still expensive to buy sensors, establish some of those hyperspectral sensors are expensive. And lack of skill, I know some of us are getting exposed to the technology, but then I think we still need to do more to make sure it gets home. How many Nigerian universities do we have that as a course, or people are learning about AI and some of these developments? Data storage and internet challenges. Can we fly drones, have GPS connection? And in some areas, some of the things we did in GRAPE, it wasn't just a government-funded. There were other parties like NASA. We collaborated with NASA to calibrate. So we were using their satellite data to adjust our field location and all that. So it involves a lot of collaborations too. So this, just to mention a few. But then the road ahead, do we just give up, or do we continue to make efforts? Public-private partnerships. Somewhere, as we're talking, we are looking at what can we give back, in what ways can we interact more, and we can build that relationship. And the last example I mentioned is an example of public and private partnership. In some instances, different government organizations and agencies were helping. NASA has that satellite, more precise data we could use. So most of our fields in the research institution were mapped. So we were able to correlate even before we develop our own calibration. They had some field indices you could use to know that, okay, NDV was just easy. So you could just use those things and then use as a proxy to whatever you are measuring, depending on the size. For grape, for breeding, it might not be important because you're looking at measuring each line in a breeding program. But then when you are looking at a higher plot size, you might be able to get some accuracy in using their data. So it might vary. There might be, let me not just say that it's like a simple one model fits all. Training, how many African scientists. We are building a network. We need to communicate more, open source platforms for wider access, and then bridging the education and gap. I mentioned that we need to integrate this into our curriculum in the universities in Africa and all that. So in summary, I think this is a technology we can capitalize upon, although I said it requires cost, but I'm thinking that compared to other costs of running a full weight chemistry for other trades, we might have advantage here. The technology we can share, we can relate, people can easily learn it. We can lead this. There are some countries that I see in the lab. Once you see somebody doing that, I can predict that that person is from one particular country. We can capitalize on some of this, and then I think is the future for agriculture. I don't want to talk about, I've had experience in industry. There are more wider application to this. People are doing inline calibrations, inline data acquisition that nobody has the time to start doing manual phenotyping. So we have opportunity to maximize on this and then utilize it to change the scope of African breeding, and then they meet the needs for more food in Africa. So while I acknowledge that this is a collaboration between different institutions, I didn't present Bayer's information, but I have to take permission to do this, and I'm currently working for Bayer, so I have to acknowledge them too. So most of these sponsored by NextGen in cassava, and then there are so many other ones maybe I've missed. Kansas State University, Cornell, some of the other experiments were done under while I was in Cornell University. So thanks for listening, and then we can ask questions, and then, but overall, I believe we have opportunities. We can collaborate, we can ask questions and build upon this, and see what we can do. Africa has unique crops we don't have here: cassava, cocoyam, crops we can't see in other regions or parts of the world that we can build upon, and then develop or mirror what has been done in other places, and develop our own area and help people in Africa. Thanks, and I'll stop sharing.
Thank you so much. Thank you so much, Dr. Ikeogu. Quickly, we have some. Let me start from the recent one, then I'll just move up.
Yeah, let me also have access to the.
Okay. Yeah, there are questions on the chat box. The first one says, how can we achieve high throughput phenotyping for measuring in situ root traits? Like I guess on the field.
Sorry, man. Throat is already dry. Let me understand the question. How can we achieve high throughput phenotyping for measuring, in situ root traits?
Yeah.
Yeah, it's possible. That's another dimension. People are looking into that. And then depending on the crop you're looking at, cassava, people started using things like, what do you call it? Wave sound. And I think that research was going on before I left Cornell. But yes, it's possible. We can think about, like what I said, is that you first look at the problem you want to solve, look at possibilities. What kind of technology do you need to adopt to solve that problem? Is possible, and we can borrow from different places. Like I said, the first device we adopted for high throughput cassava phenotyping, they told us it was actually made for mining. So but eventually we converted it, saw the use in food processing, and it's working for us. So yeah, decide the problem, and then we can go about the technology. But it's possible, it could be challenging, but then people are working, and there are different sectors you can borrow knowledge from to develop a solution for that.
All right. Yeah, thank you so much. Um, there are two others. They are not really questions, but I think they are calling for collaboration. Iwan, I guess I'm right with it. Him says, I would like to collaborate. We need fortified cassava. The other person is Obanda. Like any participant having funded project I can do for my MSc botany, I would appreciate it. So I guess these guys, if you're asking maybe for collaboration or whatever, I think you can reach out to Dr. Gochuku if that is fine. I don't know, whatever might be your specific question, you can always reach out to him directly. And I guess, Doctor, you'll be willing to like share your email if somebody's wants to kind of partner or collaborate.
Yeah, sure, definitely. Like I said, personally, some projects were happened in my career stage. I'm still growing, but then some of those because
Can you mute yourself, please? Yeah, thank you so much.
yeah, there are some of opportunities to develop, and then I see Enoch on the screen on this. I think he's raising his hand. Some of these devices, like that of Cassava, we had more devices that were procured for IITA in Ibadan. One is in Umudike, National Rural Research Institute in Umudike. And if you're in other countries, we have one in Uganda, and I don't think we eventually purchased for Ghana. But collaborations are possible. Wilfred is here. Wilfred came all the way from Uganda, Kenya, and visited Umudike and used the same device in his master's. So still possible collaborations. I know it takes costs. NextGen sponsored that. Funding is an issue that we still need to maybe is a reality we need to face, but then I believe we can find ways to use what we have on ground, and then we can expand that network. It could be an institution, somebody in the US, somebody in another. Those are, the ones I've mentioned are the ones I know. There might be some other innovations and initiatives going on across Africa that we can maximize collaboration with other people to build such technology. It can also come into data processing. You might not start from data sampling. You can utilize the data and then come through aspect of calibration, AI, machine learning, and then build calibration. That could be an area you can focus on instead of beginning from the starting from the beginning.
All right, thank you so much. I think I would call Enoch. There are like two questions that we're trying to manage time. So Enoch, I want to give you the floor so you can ask your question quickly. Thank you.
Thank you very much. Thank you, Dr. Ugo. Always been a big fan of the work you're doing on high throughput phenotyping. My question is really in line with a comment you gave about strengthening the need to strengthen collaborations among scientists in Africa to be able to exploit or leverage the existing tools, especially for phenotyping. And I just wanted to know whether you think data sharing is one of those things that can be explored. I'm asking that because you're very familiar with the process of training these equipments to predict these traits, but clearly it's a long process. So whereas the institutions might invest in these equipments but the process of training them might also be a tedious one. But I think if well advanced, maybe institutions, maybe like NRCRI, who have done maybe these robust predictions, maybe we're able to share them with the other emerging, uptakers. To me, I feel like it could be something that could fast track, adoption of these technologies. But I want to hear your take on that. Thank you.
Yeah, nice to see you again. It's been a while. Yes, we can collaborate, and I might not speak for all the institutions across board, but I see possibilities. Some of the some of the data might be public, while some others might not be public, but some have been already like published, and you can have access to them. So my take would be like there is no harm in trying. Some of the things we are doing, strengthen our collaborations. If you know where things are, and hoping we have more of these establishment or initiatives where we can share knowledge. I think African Breeders Association and all that, some of these organizations that are coming on board, those we can raise in some of those platforms. But I believe we can easily have access to data. Like I said, some of the work I did, we had to borrow data from NASA. Those are publicly available. You just download or ask for permission, and then they send them to you. So we can do the same. We can reach out to each other and then see what we can do. And yeah, but overall, I think it will be encouraged and is a way to strengthen our capacity in this direction.
Yeah, thank you so much, Enoch, for the question, and thank you for your response. I think I will be able to take this one. It says, do you have example of application of HTP, I guess it's high throughput phenotyping methods for disease detection in cassava, any other crop? In cassava or any other crop?
I showed what we did in grapes, annotation for diseases in grapes. So that is possible using computer vision, or depending on what you are looking at. We have an effort that was going on for using the same, because we are scanning the fruit. So we had a scan on the fruits and on the leaves. We're able to detect and associate some components. So a stressed material can be looking at lesser water capacity, looking at different mechanisms. It could just be plants respond in different ways. So you can base your technology in different things. So a well-stressed plant could just be less in maybe what rate will I use? So it's possible. We have done some. Like I said, I have data for a lot of them that we have not processed, but there are a lot of examples in what we have done. There is also like a collaboration with Embrapa Brazil on diseases on cassava specifically. That data has not been finished, fully published, but is also something, an effort. So we can discuss more on that if somebody is interested in that. Yeah, but the answer is yes.
Yeah, thank you so much. I guess we'll have to stop here now. I'm really sorry for questions that we cannot take here, but again, I would advise that if you have any question, you can always reach out to Dr. Ugo, and I see he's willing. Yeah, somebody's already asking for your email. He's willing to take questions. Yeah, just quickly before we wrap up, There's a post-webinar feedback on the chat box, and I'm also going to share my screen now. So please, we want you to give us feedback on what we are doing. We want to hear feedback. Every feedback it's okay. The only thing that we will not kind of encourage is no feedback. So please tell us, give us ratings about how you see this event or how you assess this event, and we can always work on your feedback to improve on subsequent events.